What's up, YouTube? This is your boy Rose Malik OG, and we're back with OG University. Let's get right into this article. So it says over the last decade of research in neurologic diseases, scientists have explored the use of human brain organoids as an alternative to mouse models. That's interesting. These self-assembled 3D tissues derived from embryonic or pluripotent stem cells more closely model the complex brain structure compared to conventional two-dimensional cultures. Until now, the engineered network proteins and molecules that give structure to the cells and brain organoids, known as extracellular matrices, often use a substance derived from mouse sarcomas called matrigels. This method suffers significant disadvantages with the relative undefined composition and batch-to-batch variability. The latest research offers a solution to overcome matrigel's weaknesses. Investigators created a novel culture method that uses an engineered extracellular matrix for human brain organoids without the presence of animal components and enhanced the neurogenesis of the brain organoids compared to previous studies. This advancement in the development of human brain organoids free of animal components will allow for significant strides in the understanding of neurodevelopmental biology, said this senior author, PhD. Scientists have long struggled to translate animal research into the clinical world, and this novel method will make it easier for translational research to make its way from the lab to the clinic. The foundational extracellular matrices of the research team's brain organoids were prepared of human biocyanic, a protein that serves as a native structure for stem cells to adhere, differentiate, and mature. They were supported by a highly porous polymer scaffold. Using proteomics, researchers found their brain organoids develop cerebral spinal fluid, a clear liquid that flows around healthy brain and spinal cords. This fluid more closely matched human adult CSF compared to the landmark study of human brain organoids developed by Matrigel. When our brains are naturally developing in the utero, they are, of course, not growing on a bed of extracellular matrices produced by mouse cancer cells, said the PhD, who was a graduate student at the University of Michigan Macromolecular Science and Engineering program at the time of the war. By putting cells in an engineered niche that most closely resembles their natural environment, we predicted we would observe differences in organoid development that more faithfully mimics what we see in nature. The success of these xenogenic free human brain organoids opens the doors for reprogramming with cells from patients with neurodegenerative diseases, says the author in the PhD. There is a possibility to take the stem cells from a patient with a condition such as ALS, Alzheimer's, and essentially build an avatar mini brain of the patients to investigate possible treatments or model how their disease will progress, said Feldman. These models would create another avenue to predict disease and study treatment Personalized levels for conditions that often vary greatly from person to person. Well, if you like talking about human brain, organoids, like, comment, subscribe. Might be able to cure ALS or Alzheimer's in the future. So let me know in the comment section how you guys feel about lab grown human brain organoids. We out.